All right, we're going to pick up here with part 4 now, talking about our neuromuscular junction. And so this is going to involve the motor neuron associated with the nervous system, as well as the junction at which it meets our skeletal muscle fiber. Okay, so obviously, in order for our muscles to contract, we need to have some type of stimulus. We need to provide it with some type of innervation. And this is going to occur initially at our neuromuscular junction. Okay, so we know that our skeletal muscle from previous lectures in different parts of this lecture is voluntary, so meaning we have conscious control over this type of muscle tissue. Now, the way that we do that is that we innervate it with a motor neuron, a lower motor neuron exiting the spinal cord, traveling out into the periphery and innervating that muscle. And so each motor neuron plus a motor fiber, or I'm sorry, a muscle fiber or a muscle cell is going to compose a single motor unit. Okay, and at this point, down at this neuromuscular junction, which is a part of this motor unit, we're going to release little vesicles of neurotransmitters, and we'll talk about what these neurotransmitters are and exactly how they act. But these neurotransmitters are these chemical messengers that are going to stimulate our muscle cell at the very beginning part of our muscle contraction. Okay, and then at each, and these are acting at our neuromuscular junction, and each muscle fiber is going to have a single neuromuscular junction. It's typically in the middle of the cell, and we'll look at a picture here. So here we see a motor neuron. Here we can see axon terminals coming down and innervating one muscle fiber, another muscle fiber, and another one here. And so each of these is a motor unit, the neuron that it is innervating, as well as the individual muscle fiber. So here's a individual motor unit. So what's happening at this neuromuscular junction? Well, we have axon terminals, which you saw in the previous image. And at this axon terminal, we have a little kind of swelling, a little region that is kind of bulbous in shape. And this is called our synaptic knob. Now, sometimes these are called synaptic bulbs, synaptic knobs, synaptic end knobs. You'll hear them referred to as different things. But it's at this point where we're going to release our neurotransmitters. So we'll also see in the next image that there is a motor end plate that is formed at the junction of where our neuron meets our muscle fiber. This is going to involve the synaptic knob that is releasing our neurotransmitters, and then the portion of the sarcolemma that is directly adjacent to that synaptic knob. Okay, it's important to note that they don't touch, but they're separated by something called a synapse. And a part of a synapse, the synapse is really referring to the whole kind of junction between one neuron or one cell and another cell. And then the synaptic cleft is specifically the space between these two cells. So we'll see that they're not touching, and the space in between is the synaptic cleft, and it's across the synaptic cleft that our neurotransmitters will travel and ultimately exert their action. So here we can see at our motor end plate, our motor end plate is involving this muscle cell. So all of this here is going to be our muscle cell. You can see the sarcolemma here. Here's our motor neuron with this synaptic knob or synaptic bulb here. This distance across this space from the sarcolemma, the muscle cell, to the synaptic knob or synaptic bulb of the axon terminal, this distance is our synaptic cleft. So you can see that denoted here. And the synaptic cleft exists all the way around. So it's not just here where it's being denoted. It's all the way over. You can see these little circles within the synaptic knob. These are going to be vesicles containing neurotransmitters. And here you can see all of these individual smaller black dots. These are representing our individual neurotransmitters. And the neurotransmitter at work at the neuromuscular junction is something called acetylcholine. Okay, so this acetylcholine, it's sometimes abbreviated capital A, capital C, lowercase h, so ACH. And then we will see that there is also an enzyme that is called acetylcholine esterase that will break up our acetylcholine after it has performed its function. Okay, so this is a good image to just to represent what exactly is going on here at the junction of our axon terminal with our synaptic knob and our muscle cell here. So Like I said, our acetylcholine is the individual neurotransmitter. These are packaged into vesicles, and we'll see exactly how they're released and in response to what. They'll bind with the sarcolemma. There are receptors on the sarcolemma of the muscle cell that allow acetylcholine to bind. And then ultimately this will be broken down by acetylcholinesterase into its constituent components. So acetic acid and choline, these two can then be reformed and then made into acetylcholine again to be used additional times. So we'll answer some questions here. What are our myofilaments? So our myofilaments are our different things within our myofibrils. So we know that we have thick and thin, actin and myosin. What is the functional unit of a skeletal muscle cell or a muscle fiber? We know that this is the sarcomere, right? And then within the sarcomere, we have it's the Z disc to Z disc or Z line to Z line, and we have those other bands and zones within that. What is a motor unit? Well, a motor unit is the individual neuron, motor neuron plus the muscle fibers. And then what neurotransmitter is released at our neuromuscular junction, and what's the enzyme that breaks it down? So the neurotransmitter is the acetylcholine. Remember, it's released from those vesicles across the synaptic cleft. And then the enzyme that breaks it down is our acetylcholine esterase enzyme. So we will talk briefly about the kind of electrical potential, electrical capabilities of muscle cells, and then we'll begin to get into the actual muscle contraction steps. So our muscle cells, just like neurons, they're very excitable. They change their voltage in response to different stimulus, and they do this by channels, ion channels, that allow different ions or electrolytes to travel either into or out of the cell. Okay, as I was saying, our cells have a electrical potential to them, meaning that they can change their electrical charge. They're excitable, and this is due to ions moving in and out of the cell through different ion channels. Now, the main ions that we talk about are sodium and potassium, but we'll also talk about calcium when we talk about our muscle cells, because calcium is very, very important and a very important ion for skeletal muscle contraction, as well as cardiac muscle contraction as well. So our primary extracellular ion, sometimes called your extracellular electrolyte, is sodium, and your primary intracellular ion or primary intracellular electrolyte is potassium. So this means that the main ion that you find inside the cell is potassium, and the main ion you find outside the cell or extracellular is sodium. Okay, now they're found on both sides. They're found both inside and outside the cell, both of these, but they're in a greater concentration either on the inside or the outside. Now, we have a net negative charge of our cell inside relative to the outside. And so what this means is we have more anions or negatively charged particles on the inside of the cell, and this makes us polarized. So polarization, polarized, meaning that two things are opposite, so a north and a south pole, they are opposite. Our cells have a polarity to them, meaning that they have a difference in charge with the inside relative to the outside. And for these cells, it's roughly negative 90 millivolts, okay? And so this picture kind of demonstrates this idea that you have potassium primarily on the inside, you have sodium primarily on the outside. Now this image isn't, it's not my favorite image, only because it only shows potassium inside and it only shows sodium outside of the cell. Whereas what I just said is that potassium you find both on the inside as well as the outside, and sodium you find both on the outside as well as the inside. Okay. Now you may be thinking that if we have potassium on the inside and it has a +1 charge, and we have sodium on the outside and it has a +1 charge, why isn't the difference between the inside and the outside net 0? No charge difference. So what you can kind of deduce from this is that there must be something else inside of the cell that contributes a negative charge, that contributes to the overall polarity of the cell, and that's exactly what we have. You have negatively charged polymers that you can see here, so things like different proteins or nucleic acids on the inside of the cell possess a net negative charge that contributes to the inside of the cell being more negative relative to the outside, so we have this polarity of roughly negative 90 millivolts. This is what we'll call our resting potential, okay? And so we will now get into the actual electrical phenomena of our cells, okay? So what's actually happening at this neuromuscular junction that is allowing this to happen, okay? So what we have is when a cell is stimulated, when our muscle cells are stimulated, we have sodium ion gates or ion channels open, and this allows sodium to move in. Now we said that sodium is primarily on the outside of the cell, and so it moves from a concentration where it is high to an area of low concentration, so it rushes in. This makes the inside of the cell more positive, and we call this process depolarization. We then have, after that happens and we reach some maximum voltage point, we have our sodium gates closed, we have our potassium ion gates open, and now allow potassium to leave. We said that potassium is primarily inside of the cell, so it goes from an area of high concentration inside to an area of low concentration outside, and this is called repolarization, okay? During this time, muscle cells cannot be stimulated again, and it's called the refractory period, and both of these things, this depolarization and repolarization, make up our action potential, okay? So that's the basis of our action potential and what we will begin to talk about here. So we talk about our negative 90 resting membrane potential. We have some stimulus, sodium rushes in, it makes it more positive on the inside. We then have sodium channels closed, potassium channels open, potassium rushes out, taking with it its positive charge, and we drop back down to negative 90 or our resting membrane potential, okay? So for our skeletal muscle, this is going to be specifically our skeletal muscle. We have this action potential that we've just discussed. It's traveling down the axon of the neuron. And these events that I just talked about here, this is traveling down the axon of a neuron. Okay, so this is what's happening in the neuron. And when we get to the end of the neuron at our axon terminal, now we're going to talk about what's happening there leading into our muscle cell. Okay, so I just want to make that clear. So when this action potential reaches our axon terminal, those ends there, what you have are voltage-gated calcium channels open. Okay, when these calcium channels open, you have lots of calcium on the outside of the synaptic terminal, the synaptic bulb. It's going to rush inward, and when it rushes inward, it actually stimulates our synaptic vesicles, those vesicles containing acetylcholine, to fuse with the end portion of the synaptic bulb, synaptic knob, and exocytose that acetylcholine into the synaptic cleft. So that image that we saw earlier, that synapse with the synaptic cleft, all of that acetylcholine is going to be dumped and exocytosed out of its vesicle into that space, into that synaptic cleft. And the stimulus to do that is the action potential arriving and stimulating the opening of voltage-gated calcium channels. Voltage gating just meaning that they open and they are responsive to changes in the voltage, right? So after we exocytose that acetylcholine into the synapse, that synaptic cleft, the acetylcholine has to go somewhere, and it's going to diffuse across the synaptic cleft and bind onto our motor end plate of our muscle fiber with its receptor, and it will bind to those receptors on the sarcolemma at the motor end plate. Okay, when acetylcholine binds here, it opens up sodium ion channels to rush into the sarcoplasm, and then potassium ions will leave the sarcoplasm. Okay, so you get the continuation of our action potential. Okay, now for this, when this acetylcholine binds to its receptor, it's actually called a ligand-gated receptor, meaning that it binds the ligand, which in this case is acetylcholine, and the receptor itself is also the channel. So whenever it binds the ligand, which is our acetylcholine, it binds it, and that binding causes the channel, the receptor, to actually open up and form a channel that allows sodium ions to rush in. Okay, so you get the continuation of the action potential that started with your neuron traveling down the axon of the neuron, and now it is within our sarcoplasm, which is the cytoplasm of our muscle cells. Okay, so now that we are at our cell, we're at our muscle cell, muscle fiber, we've transmitted the action potential to the muscle fiber. we're now going to continue this action potential down the muscle. And so it will travel along the sarcolemma, and we know that the t-tubules, also called the transverse tubules, at certain points make these invaginations down deeper into the muscle fiber. It's these t-tubules that carry the action potential down, and when they travel down, we know that on either side of the t-tubule you have the terminal cisternae of the sarcoplasmic reticulum. Remember those three things: two terminal cisternae and the t-tubule make up the muscle triad. When it travels down that t-tubule, it stimulates the opening of voltage-gated calcium channels to open. So when these calcium channels open now, you have a high concentration of calcium within the sarcoplasmic reticulum. So this calcium is going to rush out of the SR, out of the sarcoplasmic reticulum, and into the sarcoplasm of the muscle cell itself. Okay. After this calcium has left the sarcoplasmic reticulum, it has entered into the sarcoplasm, it's going to go and bind to the troponin. So if you remember, troponin was one of the regulatory proteins that we discussed earlier, along with tropomyosin. When calcium binds this troponin, it's going to shift this troponin-tropomyosin complex off of the actin or to the side of the actin, and this will expose on the actin molecule itself binding sites for the myosin head. So you remember, the myosin was the thick filament, actin was the thin filament. When this reorientation occurs and this troponin-tropomyosin complex shifts, it exposes the binding sites of myosin, and then the myosin head can bind to the actin. Now there's some more steps that we'll add in a little bit that actually make this binding possible. You have to have ATP to energize the head in order to make it form what we'll call this cross bridge and bind to the actual actin molecule itself. Okay, so here you can see, this is just kind of a walkthrough of what we talked about. We see our synaptic vesicles being exocytosed, exocytosing their acetylcholine out here. Remember, what was the stimulus for this happening? This was calcium rushing in. And so if we look in here really closely, as this acetylcholine is diffusing across the synaptic cleft, it's binding to an acetylcholine receptor, and we said that this is called a ligand-gated receptor, meaning that when it binds the acetylcholine, it actually is the channel itself. So it opens up and allows sodium to continue into the cell. You would then have potassium leave, and this action potential would continue down the sarcolemma of our muscle cell, right? And here's just a depiction after we bring in the action potential to our sarcoplasm and we continue down here. That's what these arrows are representing. We have the T-tubule, or the transverse tubule, kind of invaginate down deeper into the sarcoplasm. We stimulate the opening of voltage-gated calcium channels, and this releases calcium where it is in high concentration within the SR out into the sarcoplasm where it is in a lower concentration. This calcium then makes its way over to the troponin-tropomyosin complex and binds to the troponin. This exposes the binding site on the actin, so you can see these little green regions here. This is when on the left here, if we back up, this is the area in which troponin-tropomyosin is actually covering the actin binding sites, or the myosin head binding sites on the actin. When this reorients and shifts, you have the myosin head able to bind the myosin head binding site on the actin itself, but you have to have the presence of calcium in order for that to happen. And this process of moving the myosin head across and the head binding with the myosin head binding site on the actin, that is called cross bridge formation. So the myosin head and the myosin itself has now formed a cross bridge and is connected to our thin actin filament. So we're going to stop there, and we'll pick up with the sliding filament theory in part 5.